parler en français juste pour introduire le midi conférence qui est organisé par le comité des étudiants du CRIUGM. Donc, on a invité aujourd'hui Dr. Amanpreet Badhwar pour nous parler de son parcours, donc vraiment au niveau plus peut-être un peu personnel, donc les défis qu'elle a rencontrés dans son parcours. Aussi, Dr. Aman est artiste, donc elle va nous parler de son art et le tout en nous parlant bien sûr de ses projets de recherche et de sa passion pour son domaine. Donc, I'm going to let you begin, Dr. Aman, and after that we're going to have a session of questions and we're going to have a discussion. So, you can go.
Yep. I'm going to share my desktop first and then turn on. So do you just see mountains at the moment?
Yeah.
Okay, perfect. Okay, now you see a slide? Okay. All right. So I'm going to try this in 2 lines in French and then switch to English because I'm still very conscious about my accent. So, bonjour à tous. Aujourd'hui, je vais vous présenter mon voyage or my journey, which can be aptly described here as anything but a straight road. Okay. As most of you know, I'm an assistant professor at the University of Montreal, and I'm going to just start my discussion with a talk outline because Amel wanted me to mention so many different things that I thought it might be actually useful this time to have a talk outline because I will be taking you back and forth in time a bit. So this would give you some reference. And also, given that I'm both a neuroscientist and an artist, throughout my presentation, I will present you with artworks and tell you a bit about what those artworks that I've created means to me. So the talk outline is as follows. I will start with an introduction to the Badhwar Mind Lab. Then I'll take you back in time to my academic journey, and then I'll take you back even further to my origins. I'll talk a bit about immigrating to Canada and then my more recent works on science outreach and communication, mentorship, equity, diversity, and inclusion in academia. So I don't think anybody really hopes to start their lab in the middle of a global pandemic, but that's exactly where I found myself. I started my lab in July 2020, with pretty much everything shut down. So fingers crossed, and we'll see how it goes. But before it goes any further, I wanted to stop here and thank all the people who have joined my lab. I thank them because it takes a lot of faith to join a lab without having actually met the supervisor in person, doing a bunch of online Zoom interviews, and giving a commitment to be in the lab. And with several of my lab members now, I've even worked online, which is how we're doing most of our lab work these days. So my lab won't really be there without people who take a leap of faith and join a new lab during a pandemic. So thank you, everybody. I also wanted to thank my sponsors, people who've helped me fund my lab. And then I'll tell you next a little bit about the focus of my lab. So my lab focuses on integrating observations from brain imaging and molecular omics in the study of Alzheimer's disease and other age-related dementias with the goal of discovering new biomarkers and therapeutic targets and improving methods to speed the drug discovery process. I have several ongoing projects in my lab currently, and they mostly fall under 2 themes. Theme 1 focuses on multi-omics investigations of age-related dementia with a specific focus on disease mechanisms and prediction. So project one is looking to establish the neurochemical fingerprint of aging and age-related dementias using multimodal metabolomics. And the second project is using multi-omics approaches to distinguish dementia subtypes. And the project under theme 2 is evaluating novel blood-based biomarkers in Alzheimer's disease. So brain health is dependent on the multi-scale connectedness of brain components, as I've conveyed here in my cover for the journal Neuroimage, where I was told to put together a cover using articles in the journal articles, all addressing the brain, but ranging from molecules to brain structure to connectivity. So this Neuroimage cover is an ode to the fact that the brain, though it's greater than all its small parts, it still needs the connectedness of all its parts to work properly. And in a disease like Alzheimer's disease or other age-related neurodegenerations, which is a focus in my lab, this connectedness starts to fall apart, and we start to see symptoms. But before I go into disease, I wanted to show, in the next few slides, show how I've managed to convey brain through my art. So there'll be 3 slides on that. And they're mostly going to capture the 3 main components of the brain, which is neurons, vasculature, or the blood vessels, and glia. And together it's called the neurovascular glia unit, and it ensures proper brain functioning. So in this slide, I have done a lot of acrylic painting on large papers, mostly handmade papers, to capture neurons in the brain, both in health, which is shown on the left-hand side, and say after stroke, when the neurons have died off, but they are still coming back because the brain is very resilient. In the middle is a painting that hangs at CRIUGM, and it's basically an ode to the fact that neurons are very important in generating memories. And what this painting is really special to me, because I've ingrained in this painting pictures from my childhood and my parents' wedding, etc. So to the unknown eye, you won't really know it exists there, but to me, it was very important to have a part of me in this painting. So that's on neurons. The second slide looks at brain blood vessels. And here we have a 7T raw, a stylized image of a raw angiogram taken on 7T. And we can really see the vastness and the complexity of the brain vasculature there, which is really essential for supplying the brain with oxygen and nutrients, given that the brain is active even during rest. And something unhealthy happening in the vasculature really affects the brain and its functions. Of course, I also wanted to point to the fact that the vessels are just not long structures, but they're actually composed of proteins. And here I've taken all the proteins that I had identified during my PhD work in the brain vasculature, and collaborated with another individual to do a deep learning sort of figure with these protein networks. And finally here, I started off with a histogram, immunohistochemistry, showing the interaction between the vasculature and neurons, of course, stylized that. Just giving you a different perspective that things in the brain do not perform in isolation. In the third slide, we have the glial cells in the brain, which are very important for homeostasis and defense of the nervous system. And here we have beautiful, beautiful artwork by Ramoni Cajal, who was not only a neuroscientist, but also a great artist. And this is my interpretation of his drawings using a lot more colors. But anyways, you're allowed variations in art, so I took some artistic license. So now that I've shown you some of my brain-related artworks, I'm gonna come back to the science and specifically the multifactorial nature of Alzheimer's disease. Okay, so in Alzheimer's disease is a unique neurodegenerative disease which is being defined by deposits of amyloid beta and phospho-tau in the brain. But we know that there are multiple other factors, such as aging, genetics, and all these other factors shown in red that either work independently or together with amyloid beta and phospho-tau to then affect the neurons, the vasculature, and the innate immunity of the brain, shown in blue, which basically ends up breaking down the multi-connectedness of the brain. So what basically the same theme I was trying to show you through my artwork is the process that's happening in Alzheimer's disease. There have been many trials of disease modifying therapies in Alzheimer's disease, but they've all remained unsuccessful. And there is also growing, and most of these unsuccessful trials in AD has been attributed to intervention too late in the disease process. There's also growing consensus that lack of precision intervention targets in a multifactorial condition and heterogeneity in AD patients impact trial outcomes. Examples of heterogeneity in AD patients include heterogeneity in cognitive profile, heterogeneity in protein deposits, where only 35% diagnosed with AD show pure AD pathology upon postmortem autopsy, and which is defined as the presence of tau and amyloid protein only. So that's what I mean by pure AD deposits. And there's also heterogeneity seen in the severity of cerebrovascular abnormalities, which is present in greater than 80% of patients and can range from mild to severe. So AD biomarkers play a crucial role in addressing these key challenges. The first one being intervening earlier in the disease process with therapeutic candidates. So if you were to chart clinical trials of antibodies against amyloid, we see that from around 2012 onwards, clinical trials have been recruiting individuals in the prodromal and preclinical stages of AD. In particular, recruitment of individuals with preclinical AD relies heavily on AD biomarkers. So the preclinical are individuals who do not have any symptoms yet. So currently, biomarkers are also being developed to address the multifactorial and heterogeneous nature of AD. The Developing New Biomarkers team of the Canadian Consortium on Neurodegeneration and Aging, of which I'm a member, is combining information from different biological scales, such as brain imaging, connectomics, metabolomics, and genomics, and employing machine learning approaches to generate multi-omics AD biotypes. It is hoped that individuals with a given biotype might demonstrate similar individual clinical trajectories, and that individuals with certain biotypes might respond better to a given therapeutic candidate. So in my lab, we are adding to the CCNA roadmap by bringing in and better studying the cerebrovascular pathology of AD. And we're doing this using resting state connectomics, proteomics, and metabolomics in isolation as well as combined. And I've just have my postdoc, Natasha, who's finally arrived from the UK to Canada, who has been moving aspects of this project. I had mentioned in the previous slide that we will also be looking at metabolomics to better characterize vascular pathology. But in order to do that, we first really need to know how metabolite changes happens as the brain ages. So what is the metabolite signatures of different aging, in different ages? And this work is a really fun and exciting project that we've embarked on with Flavie Dechveri and Stephen Gia in my lab. And we are imaging individuals at a 70 MRI machine and performing MRS spectroscopy along with other imaging measures. At the same time, we are taking blood from these individuals to look at metabolites in the blood and specific vesicles that float around in the blood. So in the same individuals would have their metabolite composition or signatures in the brain and their signatures in the blood. And since we're looking at a population cohort, so from ages 20 to 69, we'll also have these signatures of how metabolite changes takes place as the brain ages. And to this, we will then add on what actually happens in disease, how the changes in age-related dementias differ from that of normal aging. So this slide is more on theme 2 of my lab, and I'll explain how that goes in a few seconds. But I'll start off by saying that in 2018, the NIAA published the ATN biomarker system, thereby shifting the definition of AD in living people from a syndromal to a biological construct comprised of amyloid beta deposition, pathogenic tau, and neurodegeneration. And this publication highlighted CSF amyloid beta 42 and amyloid PET as recognized measures of amyloid. So this is measures in CSF, PET imaging, and structural MRI, and I'll be talking about this in the rest of the slide. The CSF phospho-tau levels, so tau is here, and tau PET as measures of tau, and CSF neurofilament light levels, FDG PET, and MRI detected brain atrophy as measures of neurodegeneration. Recently, there has been a lot of excitement about blood-based biomarkers in AD, especially the amyloid beta 40/40 ratio and the tau ratios in blood. For the tau, people are looking at tau 181, 217, and 231. Once these blood biomarkers are robustly validated, they will provide a less invasive, less costly, and more accessible way to detect AD earlier. However, a major issue with blood-based biomarkers is that it is difficult to delineate the source of the biomarker. An example being the amyloid precursor protein, which is also expressed by many non-brain cells in the periphery. So this is exactly where brain-secreted extracellular vesicles, or BEVs, can provide an innovation. So a little bit on BEVs. They're released by brain cells and are nanoscale membrane vesicles containing brain-specific surface markers and molecular cargo that can cross the blood-brain barrier into blood. And isolation of BEVs from blood, a form of liquid biopsy, provides a novel, minimally invasive way to sample brain tissue. So this is what the video is showing, the release of these BEVs from the blood, from the brain, crossing into the blood and containing a lot of molecules such as proteins, RNA, etc. from the brain, quite preserved, floating around in the blood. So for this reason, BEVs are a novel blood-based biomarker category in AD. And BEVs are also being used to evaluate target engagement of AD therapeutics. And given their ability to bidirectionally cross the BBB or the blood-brain barrier, they hold promise as non-invasive technologies for delivering therapeutics across the blood-brain barrier into the brain. To date, BEVs in blood plasma or serum assessed for their biomarker potential in AD are reported to stem from 3 main components. neurons, including neuronal precursor cells, astrocytes, and the vasculature. Of these, neuron-derived BEVs have been the most investigated. Moreover, while BEVs carry a variety of molecular cargo, AD biomarker studies have tended to focus on the protein cargo of BEVs. So in my lab, we're also looking at the protein cargo of BEVs. Specifically, we're developing BEV protein panels as novel biomarkers of AD, and data from these panels can be easily integrated into a multi-omics machine learning pipeline. So I'll give you two examples of the panels that we're developing. The first example is of a mechanism-informed protein panel, where we have included proteins that are associated with one or multiple aspects of the AD pathophysiology and are known to be enriched in the brain and known to be present in the BEV. So we're getting that information from my work with the National Research Council of Canada. And in the second protein panel is designed to serve as a biomarker of cerebrovascular dysfunction in Alzheimer's disease. 15 of the 25 proteins included in this panel were selected from an initial list of 193 proteins, which I found to be altered in the levels, to have altered levels in the cerebrovasculature of mouse models of Alzheimer's disease. So these proteins were chosen both for their translatability to human and involvement in a range of pathophysiological pathways involved in cerebrovascular dysfunction in Alzheimer's disease. So some of the BEV work is being conducted, and I'll be recruiting more individuals, but for now, Sabrina Lugiani, who's been interning with me for several years now, will be joining as a master's student and furthering the BEV work. But I'm definitely looking for more individuals to work on this rather large project. Okay, so now that was the Badhwar Mind Lab, and now I take you back in time to my academic journey. I'll try to summarize it in small pictures here. I did my bachelor's degree at McGill, and from very early stages of my degree, worked at the Montreal Neurological Institute with professors Fred and Eva Anderman, they're shown in the slide there. I specifically studied the impact of genetics, in particular that of the French-Canadian founder effect, on clinical and radiological markers of neurodegeneration in the epilepsies. This work allowed me to gain an understanding on how genetics can help identify subpopulations of patients that are more amenable to therapeutic interventions. And I seem to have carried this theme throughout my research career. I went on to do a master's with Dr. Ken Duer at the Genome Quebec Innovation Center, where I applied big data analysis to genomic sequences to identify regions of chromosomal instability that can promote neurodegeneration. So the work in my master's really exposed me to big data analysis and bioinformatics on really huge data sets, which was very helpful when I started my doctoral research on mouse models of Alzheimer's disease. And I was supervised here by Dr. Edith Hamel. The kind of the unique part of my doctoral degree was, even though I was registered at McGill, it ended up being a tri-university degree, because I spent a lot of my time at the, not only at the Montreal Neurological Institute, but also at the Center for Phenogenomics in Toronto and the National Research Council in Ottawa to do different aspects of my PhD project, which looked at the role of neuronal and vascular contributions in Alzheimer's disease and how treatment affects these two compartments and whether or not it brings back cognition. Of course, all this work was done in mouse models of Alzheimer's disease. My postdoctoral fellowship was done here at CRIUGM with Dr. Pierre Ballack, but aspects of my research has also been conducted at the Max Planck Institute in Leipzig, Germany. So a really quick slide on my postdoctoral work. I studied resting state connectivity measures because it was an emerging biomarker in Alzheimer's disease, and it provides insight into the network nature of the brain so the network connectivity in the brain. During my postdoc, investigated connectivity measures in AD from 3 different perspectives, namely group level analysis, so looking at, say, Alzheimer versus control, or normal individuals, to subtype analysis, which was addressing more the heterogeneity present in Alzheimer's disease, to individual connectivity fingerprinting, to see if an individual's connectivity fingerprint gives us any more insight into how their cognitive, how it affects their cognitive performance, and if that itself would make a good biomarker. So we're moving more and more closer towards personalized medicine. There's a lot of work to be still done in this field, but that was kind of the different ideas behind the 3 main projects that I looked at. Okay, now that I've told you a little bit about my academic journey, I'll take you way back in time to my origins. I was born in India, and I spent most of my childhood in what I call the jungle called Calcutta, or now Kolkata. It's a very, very populated city, but it's also very colorful. It's interesting. It's a complete culture change if you were to go from here to there. But I felt that growing up in a different country really gave me perspectives that I might not have had I been only brought up in one country. So that's where I went to, kindergarten, etc. in. I had a family, obviously. I had my parents depicted here along with my younger brother. I had shelter. I lived in a building, in a rental apartment, and I went to an all-girls convent school in India, because most English-speaking schools in India are what they call the Anglo-Indian schools, which are really run by nuns. So it was an all-girls school, and I was actually quite thankful to have gone to such a school, because the teachers and the nuns really instilled in girls attending the school very early on that women can attain a lot of things, and nothing was really impossible. So I would say it was a very feminist school in that sense, but I'm really glad that it was, because since then life has thrown so many challenges that it was nice to get that strong background growing up. Of course, like most Indian parents, if you ask an Indian parent what you want their kids to be, it's either the blue pill or the red pill. It's an engineer or a doctor. But I had my own ideas, even from a very, very young age. And luckily for me, I had female role models to look up to, even at that time. So I wanted to be a scientist, or I wanted to be a prime minister. I couldn't quite decide, so I thought maybe I'd be both. Clearly today I'm a scientist, but I haven't quite given up on the thought that maybe you can combine science and politics and give back to that field somehow. So we'll see how that goes, but at least one of my paths were fulfilled. And then at quite an early age, I immigrated to Canada. So I immigrated to Canada as a teenager, and completely on my own, because even though I had a family, I had shelter and education in India, my family knew that in order for me to if I wanted to really have a career as a woman in India at that time, they really were not going to be able to fund it. My parents are really middle class in India. And at that time, it's changed now, I think slightly, but at the time, in order to get into a good college or university, you really have to provide a huge amount of bribe to most places to get in, or you have to be a super genius. And I don't think I was a super genius. So they thought, okay, well, she has an aunt in Canada, so why not send her there, and maybe she'll actually, you know, have the career that she wants. And it's this part of the talk that I might get a little bit emotional, so bear with me. I haven't thought about all these things for a long, long time, so it doesn't even feel like it's happened to the same person. But immigration is always a difficult process, no matter how smoothly it goes. And I've kind of tried to represent that through some of my paintings, entitled "Cause I'm leaving on a jet plane, don't know when I'll be back again." It's taken from the song, but you know, it kind of, when you're leaving the country for the first time, especially for me as a very young teenager, I really didn't know when I was going to see anybody next time around. And these two paintings called Hyperplane and The Other Lands sums up some of my feelings. Once I landed in Canada, I really felt like, you know, there were two separate spaces. We had the culture in India and the culture in Canada are really different. And as an immigrant, you really want to belong in both countries, but you end up kind of belonging in no country because you know, you're kind of in a limbo phase until it's been a while, and then you get adjusted. But the, you know, like your early years as an immigrant is quite hard. You don't have any friends. You have to make them from the start. You don't have any rituals or anything. So I found that quite hard, but I also found it quite exciting at the same time because I was discovering new things, meeting new people, etc. The second painting is called The Other Lands, and I think to a certain extent I still feel that today. So I'm going to read that out. It's not my own words, but Dame Margaret Drabble writes that the past lives on in art and memory, but it's not static. It shifts and changes as the present throws its shadow backwards. The landscape also changes, but far more slowly. It is a living link between what we were and what we have become. And then when I read that, I felt so at home. I could completely relate because, you know, sometimes all that remains of the past is the poignant memory of having belonged there. And that's the only tie that pulls us back to these lands. Because since I've immigrated to Canada, I've been back to India a few times, but the India I knew no longer exists. And I think if you grow up in India and you never leave the country, you don't notice those changes that much. But for me, every time I go back, I'm not even going back to the country I grew up in. So it's this memory of having belonged, but you also know that you'll never be able to belong there again in the same manner that you used to when you used to live there. Okay, on slightly lighter notes, I've also done several paintings of India, but more capturing the colors and textures of the land. So here we have a painting of mine of showcasing the sunrise over the Ganges River, which is shown here. This is called At the Hour Past Midnight because India gained its independence at the hour past midnight. Here we have some statues similar to the Buddha sculptures here. And then, you know, here it's Mowgli's dreams. So Mowgli is the Jungle Book character, so trying to capture the greenery of India. So there we have it. So soon after I came to Canada, I would say in a few years' time, I actually found myself in a situation where I didn't have a family, I didn't have a shelter, and maybe I had an education, but we weren't quite sure about that. Yeah, so I just, there was some family dynamics going on, and I just found myself abandoned really by my aunt's family. So these were really, really dark days. I pretty much had to support myself from that time onwards, having put myself through high school and then all the academic training going forward. So university, the different stages of training. And I really don't like, I never liked painting about this topic until 2014, because at that point I felt like I could re-examine that stage in my life. And this painting is probably the only nude that I've worked on, and it'll probably be my last. But it's called Resilience, and it's basically, the emotions here are that through adversity, betrayal, and loss, she carries on, head held high, a certain determination, a hidden strength, and a will to ride out the storm. The past is accepted but not forgotten, and is used as an engine to drive forward to better days. So though it was dark times for me, there was silver linings, and I found like a Canadian family sort of fostered me. So I had an adult presence. I also at that time worked in a multicultural facility in Ontario. So that taught me a lot. I also was lucky enough to get a job at Menasco, which designs landing gears for airplane, and I was doing a very satisfying job there. And then I came to Montreal for university, and I really was getting bored taking just purely undergraduate courses, and I wanted to do some lab work. And I was sent to the Montreal Neurological Institute to Dr. Fred and Eva Anderman. And, thank gosh that I met all these people and opportunities, because I think without these influences in my life, I really won't be where I am today. So I just want to say that even when you have those dark days, you kind of have to hang on to the silver linings, because those are usually very important. And then, dark days don't last, and life goes on and days get brighter. So after a long time, because I was on my own and I didn't have the money to go back to India. So after probably 7 or 8 years, I went back to India, and I was reunited with my family. I got to see them. This is my dad, my mom, my brother, and me. I met my now husband. I had my parents actually visit me in Canada. So here they are. And eventually I got my PhD. I should point out that my PhD degree was the only degree that my parents were there for. Every other degree, I kind of just did it on my own. And, we just didn't have the money to travel to that extent. So it was really nice to have them there for my PhD graduation. So in the last few slides, I wanted to talk about science outreach and communication, mentorship, equity, diversity, and inclusion in academia. And you can see why I would be drawn to these things, because of my rather, I guess, difficult years, moving countries and then finding myself by myself. I really started to value things that kind of give that spark at all ages to continue, like the silver linings. And I think these things really play a big part in whether somebody is happy or successful in academia. I'm a strong promoter of science outreach and communication, because at least in Canada, most of our grants come from the public taxes. And I think it's rather important to be able to communicate to the public what us scientists are doing, and not just to the adult public, but also to children, because when you communicate science to children, you kind of light in them a spark that, that they might be the scientific future that we're waiting for. So I've worked with many organizations, and here I've given examples of the OHBM, the Organization for Human Brain Mapping blog team. But I've also, with OHBM and the diversity committee at OHBM, been part of this live review for kids event with Frontiers of Young Mind. And this Alzheimer's disease paper was actually written by Sabrina Logiani, who at the time was a trainee with me. but these other papers were written by other scientists. But we got kids involved in reviewing these articles and then improving them based on what the kids understood or did not understood and their suggestions. So I thought it was a really good exercise. And these are just two examples of my science outreach and communication work. The second work I did, which I'm quite proud of, is I had the opportunity to become the chair of the student and postdoc SIG, or the special interest group at the Human Brain Mapping Society. And that's a really large international society with, maybe not so large, but at least 5,000 to 6,000 members. And at the time, they didn't really have a year-long thing for students. It was just a party that we would have, and that's all we would do. And I felt that a mentorship, an online mentorship opportunity was needed, because most people who attend these meetings, or most students who attend these meetings, are from labs that are doing okay financially. These are expensive meetings to attend. And I felt that, OHBM is an international community, and it has members where the labs might be underfunded, might be in a country where it's difficult to travel. So we needed to hook these students up and trainees up with international mentors to keep science not just limited to the rich labs in the rich countries. So that was my motive behind putting forth this initiative. And this initiative has really flourished, and it has a life of its own now. And I'm also currently the chair of the Equity, Diversity, and Inclusion Committee, which is not called that, but it's called Diversity and Inclusion Committee. But it serves the same purpose at OHBM. And again, when just a few years before I joined this committee, OHBM noticed that they had huge differences in committee members and keynote speakers as far as came to gender splitting, like how many males versus how many females. So we've been really trying to equalize that. It is a very difficult task. It doesn't happen overnight, and we have to keep pushing for that. But this committee has since diverged out to other avenues, and is now organizing inter-organization dialogue to improve EDI in academia. And the last two slides, I believe, I wanted to point out that EDI initiative is not just important for adult academics, but it should really start at a very young age, because from this publication in 2018, there's very few children's characters who are American Indian, Latinx, Asian, Pacific, African, etc. So these, if you come from an underrepresented minority, you don't really have role models that are like you that you can look up to. So one of the things that we did with the Brain Art special interest group at the Human Brain Mapping meeting is task an artist, Katie Alexander, with representing neuroscientists who were normally underrepresented and minority neuroscientists into superheroes, and give this opportunity to kids to have somebody to look up to and want to become like them, because I think that's very important for the future generation to have representation, because if you can't see yourself in a role, you're less likely to engage in that role. So that brings me to the end of my talk, and I kind of just wanted to end on my last slide, which first of all, thanks everybody for listening, but I wanted to quote this saying, this writing from Jhumpa Lahiri, who's an Indian American, East Indian American writer. And she says that still there are times I'm bewildered by each mile I have traveled, each meal I have eaten, each person I have known, each room in which I have slept. As ordinary as it all appears, there are times when it is beyond my imagination. And I kind of feel a lot this way when I was making this talk. So thank you for the opportunity for giving me the FaceTime to give this talk. So thank you.
Thank you very much, Dr. Haman. Really touching and really interesting presentation. I'm really happy to have invited you, because it's rare that we have the opportunity to have these kinds of presentations, so personal and so touching. So thank you very much. Before asking, I have some questions, but before going and asking some questions, I would like to see if anyone would like to start the conversation, if anyone has some questions. Est-ce que vous avez des questions pour Dr. Haman? Oui, Natacha, tu peux y aller.
Hi, bonjour. Aman, that was such a great talk, thank you. My question, or one question I had, was actually a bit more about your artistic background, because your academic work is obviously very scientific, and I wondered if you actually have any formal art training, or if it's all just kind of self-taught.
It's a self-taught. I tried taking an art course and I hated it. I think as far as art and cooking goes, I hate following recipes. So, yeah.
That's interesting. Thank you.
Yeah, really interesting. I was wondering, how are you able to manage art and doing, you know, like an academic career, successful academic career in the meantime? I'm really interested in arts. I do art since I was a little child, but seeing that you can go that far with your ideas, it's really inspiring. And actually, how do you manage to do all that in the meantime? I mean, it's a lot.
I guess you start by giving people deadlines as opposed to them giving you deadlines. So if I'm asked to do something, I'll be very honest and say no if I can't deliver. Mm-hmm. I do art because for me it's a way of relaxation. So it doesn't seem like work to me. It actually helps me turn off the science, because otherwise my mind is always going around and trying to solve one scientific problem, or how to write a grant, or how to write a paper. It just is never ending. So art and the other activities that I do, such as cooking or the outdoors, is kind of my only way to turn off the scientific process. And so I find it quite liberating, and it's something that I have to do, or just like, you know, drown myself in too much science.
And I was wondering also, I don't know if somebody else do have a question, or if someone wants to say something. One of my question was, Mélanie, go ahead. On t'entend pas, Mélanie?
Do you hear me now?
Yeah.
Okay. Yeah, thank you. It was just another question extending on that art topic. I was wondering if, as you mentioned, you do it as a relaxing activity, but can you expand more on the time that it takes in your day or in your week, just to see how much hope I can put into doing so myself too? Thank you.
Good question.
Well, yeah, I wish I could say I do it every day, and I don't. I try to do art every weekend, and I guess to a certain extent, like for me, if I have a project, then I'm doing art every day. If it's just for the sake of using colors, it might not happen so much. But you know, once I get an idea, like most of the exhibitions that I've done are very, they're oriented around different aspects of memory. So I just, once I find a topic that I want on my artworks to reflect, it's quite easy for me to incorporate that into the day, and you know, do a little bit at a time. My setup is very weird. I've tried having a studio, and I've tried renting an art studio, and I found I never really did any art because it was such a big effort to dress up and go to the studio. So my studio is, you know, the floor next to my kitchen. or the floor next to my computer. I do my artwork on the floor, by the way, and I do a lot of layered work in my artwork. So I need time for things to dry. So it's for me. I could be writing a paper. I'll take a 10-minute break, go apply a layer, do some stuff, then come back, continue with my paper, or go cook food, go do some exercise. So it's really like woven into my day. I don't set aside a time. It's like how I do everything else. I just kind of do things in parallel, and I am not the kind of person who can sit in front of the computer for hours. I have to take breaks, and I just use my break to do art and other things I like. So
Amazing. I think Nadia have a question.
Hey, Aman.
hello.
It's amazing having having you in this talk today. I must say, thank you so much for this moving and inspirational talk. I had no idea there were so many things behind you as a woman, an artist, a scientist, and it's actually an honor to know you. And I must also point out that people have to know that you're also very generous, kind, and very humble person. You have accepted a lot of things, and also last year when you were so busy, with your new position and everything, you also accepted to do some art for the bulletin of the CRIUGM, which is on the website. On our website, when you see, you have Aman's artwork there. It's on the front page of this new edition of the Bulletin d'Information. So thank you so much for that. I wanted to know if do you consider yourself more like an artist or a scientist, or is there anything that comes first, or it's they're just mingled with each other?
I don't think there's any delineation in my head. I mean, I do a lot of figures when I'm writing a paper, and that's very art inspired for me. And I want to do more of really cool scientific figures in the future. And most of the students in my lab are really into, especially Natasha and others, are really into mingling science and art and coming up with new ways to represent a scientific happening. I'm chair of the Brain Art Special Interest Group at the Human Brain Mapping meeting or organization, and there the goal is really to use art as a new way of showcasing science, both for scientific representation, but also to speak to have a dialogue with the public, so if you were to ask me, I don't really see myself as one or the other. These are the things I like, these are the things I do. I mean, other people play music, doesn't mean they have to choose between music or being a scientist. So I do art.
Okay, thanks.
I have maybe a last question before ending the conference. What are the advices you can give to someone who is starting, as an example, as me, their PhD and being part, for example, a woman from a minority here in the research in the academia? Do you have like highlights or advices to give? Especially you started a lab in the pandemic. I mean, you've been through much of challenges, so maybe you have words to say about this.
Well, I mean, as unpleasant as it sounds, pretty much every woman joining my lab, I do tell them that it is harder for a woman in academia. It's expected of us to give 150% or 200% compared to men of work that goes for us to be in any position, really. By being part of different committees, me and others, and that also involves a lot of men, are trying to change that quite actively. So I'm really happy to see that work happening. But as a woman, I would give you the advice is, you you are going to see things that just doesn't feel right in your guts, or people, you might come across a situation or maybe a comment that doesn't quite sit right. And I think the gut instinct is to just shut up about it and somehow make that our problem. You know, like we say we must have done something wrong, and that's why we got that comment, you know. I think my advice to any young woman would be like, no, maybe process it, take the time to process it, but don't own it. That comment was coming from somebody else, or that reviewers whatever impression of you was that reviewer's impression of you, and it goes to both men and women, really, not just women. Just keep trying. I think if you really want to be an academic, you have to be resilient, and you have to persevere, but you cannot become bitter at the same time. And if you do get the opportunity to become a PI or a professor, you have to pay, you have to be the change. You can't expect the system to change for you. You have to create that change. You have to want more. So that would be my advice.
Thank you very much, Dr. Haman. Thank you very much. I think we can end this meeting because I think people have other meetings and they are going for work. Thank you very much for the great talk. Very, very interesting, inspiring, touching. I wasn't expecting that much, so I'm very, very happy and grateful. And looking forward to see you maybe in presence one day at the CRIUGM. So thank you everybody to attend this meeting, and have a good afternoon, everybody.
Thank you for the opportunity. Take care. Bye. Merci.